Hello, everyone. Welcome to our webinar today on how to develop the ground truth for a deep learning AI model with speakers Dr. Aleksandra Zuraw from Digital Pathology Place and Dr. Lindsey Smith from Aiforia. All participants' microphones are muted throughout the webinar. If you have any issues, please send a message through the chat function. We will also be taking questions at the end, and you can submit these at any time through the questions function. I will now pass over to Dr. Aleksandra Zuraw. Thank you again for joining.
Thank you, Christine. Welcome, everyone. Good morning and good afternoon. And first, big thank you for signing up for this webinar and for showing up, because we know there is a lot of stuff out there because digital pathology is booming. So we want to thank you first for your attention and for showing up here. I'm Aleksandra Zuraw. I'm a veterinary pathologist, and I'm also a publisher at the Digital Pathology Place blog and podcast. And my first encounter with Aiforia was at the Pathology Vision Conference in 2019, where I heard Tom Westerling-Boy. He is the scientific strategy manager for Aiforia, scientific strategy person. And he was talking about obviously the software, but then he was talking about the validation, and something that convinced me that this is real and these are real people and that they're not kind of trying to make it more beautiful than it is, was where his words about validation, that you may the capabilities for validation are there in the software, but nobody's going to do the validation for you. We're not going to talk about validation, but just wanted to tell you that this is what kind of made me understand that these people are real and it's cool to work with them. And then I joined Charles River Laboratories, which happened to be a company that uses Aiforia. And this is where I met Lindsey. Lindsey was my trainer for the software. So everything I know and did in this software is because she trained me. So you are in good hands. So I'm gonna, Lindsey, let you introduce yourself and tell a couple of words about Aiforia as well.
Yeah, thank you, Alex. So like Alex said, and Christine, my name is Lindsey Smith. My background really is in neuroscience and understanding electrical circuits in the brain, but I did transition from bench research, and now I work at Aiforia, where I actually help people build AI models to expedite their data analysis. So Aiforia actually stands for Artificial Intelligence for Image Analysis. So we have a ton of use cases, previous webinars that you might want to see. You can access all of that from our website, and you can also book a demo. So we believe that seeing is believing, and so if you want a proof of concept demo on your own images, you want to see how, if this is going to work, we'd be more than happy to set that up. So there actually is a button on our website that will take you straight to registering for that, and it's free to do so. But yeah, I'm very excited today. Today's topics are absolutely critical to actually getting an AI model started, which would be, how do you think about, so what is a ground truth, number one? So how do we think about this? How do we define it? Is it this static thing? Does it evolve over time? And then also, how do you approach annotating that input data to teach the neural net that ground truth that you're trying to teach it? So there's a lot of critical things that we're going to go over today that are absolutely important to getting your AI model to work, not only efficiently, but also quickly. So there's some things that you can do that we're going to try and help you out with today and teach you about. There's some things that you can do and know up front that are really going to expedite your AI model development process so that you can get closer to actually getting that data out and having the computer automate your image analysis, basically, so that you can press a button, walk away, do more experiments and come back, and then the computer's basically doing all the hard calculating for you. But with that, I'm going to hand it back over to Alex, and we can go ahead and jump into all of our topics for today.
Okay, thank you. Let's see. Yes. So, like Lindsey said, our ground truth is what we're going to be talking about. Annotations for deep learning model development is our ground truth if we're talking about supervised deep learning model development. So we're going to talk about a couple of principles that are crucial to have quality ground truth, and as a consequence, quality and robust deep learning models. So first principle is keep your classes clean, and we're going to talk about what the class is and how to keep the classes clean. And the format of today's webinar is I'm going to tell you about the concept, and then we're going to switch to the Aiforia platform, and Lindsey is going to show you the concept in action in Aiforia. Then we're going to talk about why the size of annotations matters, and what are the numbers or the ballparks we are working with regarding the number of annotations that we have to provide for a deep learning model. We are gonna talk about how to correct our annotations, because being wrong is okay, but staying wrong is not. So there are strategies and approaches towards that. And at the end, we're gonna touch about something very important. It's a little bit beyond the scope of this webinar, but we're gonna point you to the resources. Why no one size fits all in pathology problems, meaning that different pathology problems require different image analysis solutions. And along the way, we're gonna show you how we do different types of annotations for those different solutions. So I have been annotating since 2016. Let's say I have 5 years of annotation experience, but it started with classical machine learning. And what was needed then at that time, as you see on the slide, was manual delineation of region of analysis. So if someone wanted to do analysis in the tumor tissue, we would annotate manually. I would go and annotate manually where the tumor was, so that we would only have analysis from the tumoral tissue. So pathologists or trained experts would do that. And then science, tumor science or tumor understanding and immune oncology, because we were mostly doing this for immune oncology, evolved. And there was a need for more and more granular annotations. So not only the tumor mass, but we had to annotate tumor invasive margin. And then at some point, there were requests for annotating manually all the tumor epithelial cells and tumor stroma, and then to exclude necrosis. So quickly, it became not feasible for a human to do it. Regardless, how much time, it was just not feasible to annotate whole slide images in that way. But at the same time, a small little light at the end of the tunnel was appeared. My colleagues, my image analysis colleagues, started using random forest algorithms, so other ways of machine learning, not just the hard thresholds, and they were using it for tumor stroma separation. And why this light was small? Well, for some images it worked perfectly, and for some images it didn't work at all. So I'm like, okay, does it work or does it suck? I don't know. But then deep learning came. Deep learning happened and became more and more prevalent in the image analysis world. And the way the meaning of the annotation changed. Now we didn't have to delineate the area for analysis. We were giving examples, what we wanted the model to learn, what we wanted to detect on unseen images. So from this type of annotations of delineation of the tumor or whatever the structure was, we moved to a different type of annotations. And this is what we're going to be talking about. Annotations for deep learning, they differ significantly from the classical ones. So our first principle is to keep the classes clean. What is a class? Class is basically what we want to detect in this image, like the simplest words, what we want to detect in this image. And in this image, you have this annotation here, annotation layers. There's one layer. We want to detect the tumor, but in reality, we want to detect the tumor and the non-tumor. So we have two classes here. And we need to keep, when we annotate, we give example of those classes, and we need to keep those classes clean. So if we zoom in into one of those tumors, this is a mouse model, mouse tumor model, and the dark areas are tumor cells. And when we zoom in into this tumor, you see that it's no longer the whole thing has to be delineated. I have a piece of this tumor delineated, but it is just those tumor cells are within my annotation. And now a little quiz, find the differences. So the difference here is kind of obvious. I included a little bit of the outside of the tumor in this annotation. And this is what I mean, that I have to keep my classes clean. In the first one, I kept my class clean. It's only the class of interest. In the second one, I included the background class, the non-tumor class, in my tumor annotation. This is not what we want to do. This is what we want to do, and this other one is not what we want to do. And you can see that, when you, depending on the magnification, depending on, how fast you want to do it, it's easy to make this mistake. So if you do not remember anything of this webinar, and that's the only thing you're gonna go and work with, I will consider it a success. And we can see it in action now. Lindsey's going to show us how it works on the platform on Aiforia.
All right, thanks, Alex. And so what I'm doing here is I'm going into our cloud-based platform. So we have been cloud-based since inception, 2013. We've built hundreds, if not thousands, of AI models at this point and processed over 1 million images. All you really need to get started here, at least with Aiforia, is an internet connection and a browser. So I've gone directly into our cloud. I'm just using Google Chrome here. And I want to show you that same exact image that you were looking at, this mouth lung tumor. And what I'm doing here is I'm actually entering the AI model development portion of the platform. And so what you see is when I click on the image, I'm going into the platform, and you see all of these black contours and these red contours. So I'm going to kind of explain what that means, and we're going to give actual examples in real time of how you would annotate these features. So what's really important here is that we have, okay, so like Alex said, this is technically a 2-class problem, but what we're trying to get the AI model to learn is to recognize tumor. So those are all of our red annotations. So something like here. So let's do this from scratch. We draw basically an annotation like this. It doesn't have to be big. So this is going to mark true positives. But the way that we actually get the computer to go search for this region that we've just annotated is we have to provide something called a training region. So here I'm just going to provide this black contour. So basically everything inside of the black contour is a true positive for tumor. But what's important is that we also teach the computer what's a true negative. And a true negative is going to be your glass slide. It's also going to be any tissue that does not contain tumor, so non-tumor containing tissue. It's just as important to show the computer true positives as it is true negatives. And so as far as keeping the classes clean, this is very important. And so what can happen sometimes is that if people are too zoomed out when they annotate, you're really not picking out the fine details of the border of the annotation. For instance, maybe if I'm zoomed out to 500 micrometers here, zoom level, if I annotate like this, at the outset, it kind of looks like I'm catching most of the border. But if I zoom in, I can actually see that I've made quite a few mistakes. I've included a lot of pixels for non-tumor tissue within my tumor annotation. So these are really, really easy to correct. And we'll talk in a little bit how the computer is going to find these annotation mistakes for us so that we can correct them more easily. Again, the last thing you do is you provide a training region. So within the single training region, I'm doing 3 things here. I'm showing a true positive for tumor, a true negative for non-tumor tissue, as well as the interface between these 2 things. And so this is an important concept, is that because this is supervised learning, you're wanting to show the computer different ways in which the tumor can look, the non-tumor looks, as well as what happens when they kind of meet each other. And so I usually recommend that you zoom in quite a lot, and you'll see actually quite how small the physical annotations are. You never annotate a whole slide. We'll get to that in a minute. But here it's important to dedicate your tumor annotations just to tumor, and also show the computer examples of a true negative here, which again is glass slide or any non-tumor containing tissue like this. So with that, I'm going to hand it back over to Alex, and we'll go back into our presentation with the PowerPoint, and we'll come back to this in a minute.
Lindsey already said that we do not annotate the full slide, and the size of the annotation matters. Small to medium annotations are less prone to error, first of all, and second, they're faster to draw. Because to keep them nice and keep the classes clean, you do need to zoom in sometimes. So to be faster and still do it at the level of detail that is required, we stick to small and medium annotations. Another thing is what Lindsey also already mentioned, what she did in the last training region. You need to provide context. You need to annotate neighbor classes. So in this case, we had the true positives for tumor and true negatives for non-tumor. And if we only have them like away from each other, we don't provide context. We provide context when we include borders in our training region. And another thing is to include the feature variability within your annotations. So in this case, our tumor annotation or tumor class is relatively homogeneous. Our true positive is more or less homogeneous, but our true negatives are not. So we have the true negative that is just glass, just white. Then we have true negative that has higher density of dark pixels. And we have true negative that has lower density of those pixels. So this is the full variability of what our non-tumor is. So we have to include the feature variability within our annotations. And this kind of dictates the size. First, not too big because we want to keep the classes clean. Second, we need to provide context. And third, we must include the full variability of the class we're annotating. And with that, I give it back to Lindsey to see it in action.
All right, thank you, Alex. And so I think we just did this previously. Oh, by the way, there's some keyboard shortcuts if you ever make a mistake like that. And again, I drew this a little bit big because we were zoomed out when I was giving that example, but really you'll want to make your annotations much more reasonably sized. So let's go look at some feature variability, not only in our true positive classes, but our true negative. So here, again, there's some keyboard shortcuts. We can kind of toggle on and off our annotations. If I move into this region, I can see that we have a pretty purple stain. It's very densely stained here, but we also have these other features like some tissue shearing or some more white spaces or fat globules here. That might look a little bit different. You might have more or less of these features in other areas, for instance, and so we've included that here in this annotation. But then what if you get to a region where it's not this gigantic massive tumor? What if you have much smaller portions? And so you might want the computer to be able to figure out and analyze these portions as well, where you have other tumor classes. But you also need to define kind of what background is as well. So background, like Alex says, looks very different along the slide. So in some places you have higher density pixels, some places you have lower density. And so it's really important when you're placing these annotations that you don't mix the two classes. So here we're just going to mark a true negative, and that's going to look a little bit different in stain than some of the other regions on the slide. So up here we see we've also marked a true negative. We have a little bit more red blood cell clusters in here is almost what that looks like. And so you need to really just include all of the feature variability. And like Alex said, also include borders. So the border, what we saw previously was a border between here, for instance, the tumor and non-tumor containing tissue. But we have to keep in mind that feature variability includes the glass slide here. The computer, we see it and we kind of ignore it, right, as the human, because we know it's unimportant, but the computer doesn't know that. So we actually have to show the computer examples of the interface between anytime a true positive happens, and then give it just variable examples. And again, you don't have to annotate the whole slide. You don't even have to give it too many annotations for it to start figuring out this task. You'll actually, but you do want to show it, the border between these. And so we'll come in a minute, but just to give you an overview, before we added those annotations, we trained a neural net, really small level of training, and just to show you how the computer can perform, even on this small of annotations. The annotations were purposely placed to capture the feature variability, not only in the true positives, but true negatives. But when you do that, the computer can actually learn quite a bit in a very short amount of time. So my internet might be a little slow here, so I apologize. Here we go. So what I'm doing here is I'm just activating the analysis results. So this particular neural net, I think, was only trained for maybe 10 iterations, which is incredibly low. But you can actually see, compared to the annotations that were given as input data, it's doing quite a good job of picking up on what looks like those darker tumor pixels. So I'm going to hand it back over to Alex. We're going to talk about how to correct incorrect annotations, things like that, and a laundry list of other things to go over today. But this was just a good example of including that feature variability allows your model to generalize across tissue that maybe it was never trained on, which is exactly what we want. So with that, I'm going to bring back up our PowerPoint and hand the presentation back over to Alex.
So at the beginning of my annotation career for deep learning, I would go in and look at the slide and just annotate whatever I saw in the slide in the proportions I saw in the slide. So I didn't even like ask myself the question how many annotations I need. I would look at the slide and annotate what I saw there, right? And so in this case, this is a blood smear from a patient that is infected with malaria. These are Plasmodium. Those little dots in the red blood cells are Plasmodium malaria parasites. So in this case, I would go in, annotate plenty of normal red blood cells, a couple of those infected, and even less white blood cells, because they're less, right? And I didn't even think about it. I would just do it. And the thing I was not aware of is that this is not how how it works for deep learning. In deep learning, there is, in general, in medical data sets, we have the problem that is called class imbalance. What does that mean? That most of the things we are looking at in images in this particular image would be normal. There's a lot more normal than abnormal, and this counts for populations. When we are comparing diseased to healthy, there's more healthy individuals than diseased. This counts for radiology images. This counts on different levels. Basically, in medicine, the data sets are not, there is no even distribution in data sets. So this is called class imbalance, as I already mentioned. So here we have this red fish. If we're looking for red fish and there is so many more other fish, we have to mitigate this problem, this class imbalance. And there are different ways to mitigate it, but because our webinar is about annotations, we're gonna tackle how we can do this with annotations. And basically for supervised deep learning, this is our ground truth. So basically what we feed into the system, this is the quality we're gonna have. And we have to take care of all the problems. So one way we can take care of this is what is so-called oversampling of the minority class. Means I go in and I find the minority class, which would be, in the case of our example, the infected red blood cells, and I annotate more of it than I actually see in my field of view. I go actively look for these. And what I do then, I kind of match the number of annotations that I did for the minority class with the annotations of the majority class. In this case, that would be the normal red blood cells. And obviously, we have to take care to annotate all the appearances of the normals as well, because they don't look all the same, and all the appearances of the abnormal ones. But basically, I do not do it proportionally. I do not do it proportionally to what I see in the image, because the neural net has to see enough examples of all the classes. And if we over provide it with one type of examples, it's going to focus more on this. And there are different ways to mitigate this. And Lindsey is going to touch on some of them. They are on the back end of developing the deep learning model, but annotations is what we can do up front.
Right,
so let's see this in action.
Yes, thank you, Alex. Okay, so let's actually go in here to this project that we were just looking at. So the red blood cell of the smears with the malaria detector. So what we have here, this is a slightly more complicated AI model because it contains more classes. And so before we saw tumor and non-tumor, that was pretty easy and straightforward, right? Just to recognize this and label this one class. But what if you have a neural net that you're trying to train to do 3 simultaneous tasks? right? And so as Alex said, we're talking about class imbalance, how to annotate appropriately. So there's a couple of tips and tricks that I personally tell people that I'm training on the platform. One, Alex was completely right. You're gonna actually give multiple examples of what these things are. So this, for instance, is an object detector. It's a different style of annotation. So this actually uses a predefined shape. You can batch edit the size for each class. We do recommend that you keep the size for a single class consistent. So what you're doing there is you're simplifying the job for the neural net. It's completely okay if your object itself is a little bit bigger or a little bit smaller than your object detector. That's not what's important. What's important is that you center your annotation. So for instance, if you annotate these two that are centered as red blood cells, and then all of a sudden you're giving the neural net this type of information, for instance, what's happening is that the neural net's really confused. It's trying to figure out, because the main focus of the inside of this object detector is now on the division between the red blood cell and the background. So really just the style of annotation can be very important here. So not being sloppy is important. Also, everything that you're providing to the neural net should be labeled. So for instance, here, I have 6 events that are occurring. Each one has its own unique shape. Some are a little bit larger than the object detector, some smaller. I'm keeping the object size consistent between these because it's basically reducing the amount of work that the neural net has to do to figure out the task. If we all of a sudden have different size object detectors, for instance, like this, the computer thinks that is an additional class. Basically, the size difference is an additional class. So here, including the feature variability is important, but also making sure that every piece of information that you feed into the neural net is properly labeled. So oftentimes this is a mistake that's quite human to do, is to accidentally leave something out or accidentally not annotate something in a training region. So what we call that is a direct conflict in ground truth. Not only are we telling the computer to learn all of these 5 events as red blood cells, and these are the variable ways in which it presents itself, but we're also asking the neural net at the same exact time, because remember, this training region marks true positives, anything that's labeled with an annotation, but also marks true negatives. And so we have to keep that in mind here, that you want to make sure that you have everything properly labeled. It's really easy to update it. You either add the annotation or the computer is going to find this for you. So there's a couple of ways to go about that. But also, at least in our platform, we have a patented active learning tool that's going to go find low confidence regions, and it's going to help you keep from oversampling or undersampling certain areas of your slide. And what we call that is the annotation assistant. So I just gave you examples here of only red blood cells, but let's go see if we activate the annotation assistant, how it's going to show us. So what the annotation assistant does is it's automatically for you, it's going to do a whole slide image analysis, and the computer is going to take you to regions based on the last training round. It's going to take you to regions where it wants more information before you train again, and it's actually going to suggest annotations. So here, I see that the training region is this very square box. This is the computer-generated training region, and it's found here based on the last training, which I purposefully fed in inaccurate data for later purposes in this talk. But what you can see is that it's mostly doing a good job of getting the object detectors centered. You do have the option to update these annotations, for instance. So we see that most everything's labeled, but I believe that these two that it guessed as an infected red blood cell, which should have the malaria parasite like you see down here, it actually doesn't have that. So what we need to do is update the annotation. It's really easy to do. You just remove those. If the computer was wrong in the beginning, you just update it. And the reason, what we're doing is we're having the computer basically take us directly to where information is needed most. In this way, you save time, and you're not wasting your own time just going through every single analysis result visually to figure out where are my annotations most important, and how do I prevent this over and undersampling issue. So here, again, everything inside of the training region should be labeled. Even if it's half a cell, you can actually change the size of the training region if you don't want to relabel everything. And you can also increase it. Maybe I know that it did a bad job of picking out those infected red blood cells earlier, so I might actually decide to include this example down here, just because if it's really my general rule of thumb that I teach everyone is if it's wherever the neural net failed in the last training round, wherever the AI model failed, that's exactly where you need to add a new annotation. So in this way, I've corrected the false the false positive guesses here for infected red blood cells. I've given it a true positive example of infected red blood cell, and I've gone ahead and labeled all the false negative actual red blood cells in green that were initially missed by the last round of training. So once you accept this, this basically becomes a new annotation that goes into the next round of training, and you can be pretty confident that it's going into a region exactly where the computer wanted more information. It's actually really nice that it shows you this. So this can go on to infinity. It's going to continue making suggestions. My recommendation usually is use this for a few rounds. What's nice is if it gives you sort of a bogus recommendation and you don't want to have to re-annotate it, you can also ignore it. That does not change your next suggested annotation. And I think the simplest way to think about this particular tool, at least for me, is I kind of consider it the spell check of your AI model development process. It's going to physically go into your slides and it's going to find regions where there's low confidence and it's going to suggest a correction, right? So it's almost like the spell check, and you can be confident that your final sort of AI model or document is going to be better off having used this tool in some cases. So that's the annotation assistant. You can change the training region size, you can change the objects if it got it correct or incorrect. And then the more times you do this iterative process of adding a couple of annotations, running a slightly longer training, allowing the neural net a little bit more time to learn, these suggestions are going to get better and better over time. And then one last thing as well. So annotation numbers are important, not only per class and undersampling or oversampling, but it's also important that you have enough examples of the feature variability that you're looking at. So for instance, but sometimes they can actually take quite a while to generate those results. So here I'm just activating these results. This particular short training, the AI model found almost 5,000 infected red blood cell events, almost 50,000 just red blood cells, and 16 white blood cells based on the last training. So annotation number, annotation style, and number per class is important, but also you want to make sure that you get that feature variability up. So nowadays we actually have a tool. Let's say I know that the AI model is already doing a good job here. I haven't annotated in any of these red blood cell examples, for instance, infected or non-infected, but I do want to use those as examples of the variable ways in which it looks before the next training round. What I could do is actually use the converter tool here, and I'm going to go from 42 annotations on this slide. We use the converter tool. Usually it's pretty fast. You can double, triple, quadruple your annotations pretty quickly using this. So here, kind of hanging in a little bit because my internet's slow, but here we go. So I went just there from, let's see, 42 to 68 annotations, and all of these now have been converted into actual annotations. So that's how you can increase that feature variability that you're showing. I think we're gonna go back to Alex now.
Lindsey, one question. What is the ballpark of the numbers, like the total numbers of annotation to have something more or less robust? I mean, like you said, we can go into infinity, but that's not the point. Something that, you know, is good enough.
Yeah. That's a really good question. That's often what people, everyone asks. So every AI model is a little bit different. So because the task is different, but in general, what we've found over time is that for semantic segmentation, like the tumor model that we saw earlier, so when you're getting the whole region of an area, that's a different style of annotation. It's a different style of neural net. And so like that, we usually recommend hundreds of annotations, and they can be just as small as you saw. They don't have to be these gigantic. You don't have to cover a whole slide. You just want to spread out those hundreds across the feature variability that you see in your data set that you want to analyze. Same thing applies for object detectors. Object detectors work a little bit different. We do recommend about 1,000 per class, but sometimes if the neural net's already really good and the images are high quality, sometimes it can take less than that. But as I showed just here, the computer is going to show you where your annotations are needed, and we also have this tool that you can double, triple, quadruple your annotations very quickly. I was working with someone yesterday who was adding 1,000 annotations a day with the converter tool just because she's gotten so adept at using the platform. So it really does not take much time at all. Like 1,000 might sound like a lot, but it's really not. And we've built in these tools to try and be as user-friendly as possible and really have the computer, we try and have the computer do in the platform do most of the work for you, right? You just have to be consistent in how you're labeling your ground truth. You can't label an infected red blood cell as an uninfected red blood cell one day, and then label it as an infected red blood cell the next day. You just have to be very consistent about showing the computer the correct examples, non-conflicting examples of what your ground truth is.
Mm-hmm.
But yeah, so that's a good question. We've say in the hundreds for semantic segmentation, probably closer to 1,000 at minimum for an object detector.
Okay.
So I'm going to switch back to you now.
Okay. So being wrong is okay, but staying wrong is not. And Lindsey already showed a little bit how you can correct things. And this was a pretty simple problem because it's obvious where the infected cell is, and it's obvious which one is not infected. And there are projects where it's not so obvious, and you still have to stay consistent at the end. So at the beginning of my annotation career, you remember it was just for machine learning, classical machine learning with feature definitions. I would train people to annotate. I would provide trainings, extensive trainings to my colleagues so that we could scale up our annotations. So, well, as much as you can scale up something that you have to do manually for each slide. But we had a larger team, and it was possible because there were limited number of things that we were annotating. We were annotating the tumor mass, so I would train how a tumor mass looks. Then we would annotate immunohistochemistry stained cells. They were mostly stained with DAB, with the brown pigment for IHC. So that was also easy. Immune cells are round and brown. I was able to train my colleagues on that. I was proud of my trainings, and I was proud that I taught people and we could do it together. And then deep learning came again, the deep learning, the game changer, and the number of problems that could be now solved with this new way of doing image analysis increased exponentially. So it was no longer possible to train people on the annotations for every single class that we were annotating. So the rules changed, and it took me a couple of projects to realize that the rules changed, and I would have spared myself a couple of arguments in meeting where I thought, no, this is the way you should annotate and you should agree at the beginning and then stick to it. It's just not the case with deep learning. And it's okay because you don't have to annotate correctly from the get-go, but you should not stay wrong. So for deep learning, creating a good ground truth set is an iterative process, like we've seen in what Lindsey just showed. And this iterative process consists of correcting incorrect annotations, like we just did, and of adding more annotations in the challenging regions. So this is an iterative process, and you basically communicate with the model. It's not anymore about, oh, these are our rules, we stick to the rules no matter what. Because if you annotate in one way, and then you see that the model is not learning well from that, you might need to change the way of how you annotate. Or let's say, you're annotating in a scientifically correct way. Like on this example here, most of what I annotated is tumor, right? And often you don't realize that there is a mistake, and then you have to go back, and especially when you're working with other people who are helping you, it's kind of a team effort. Team consists of the people who are annotating with you, and the model is part of the team because it gives you feedback constantly. You start with few, you see how it performs. You can use the tools, which is fantastic because otherwise you would have to like look for this on your own. And this process of correcting is called sample mining. It can be either positive mining or negative mining, where you mine for the classes that are true positives, true negatives, performing well, or where you also adjust the number of your samples. But basically, this is an integral part of a deep learning model development process. And I'm happy I can share this message with you because as I said, it took me a couple of projects to figure out that it's a different way of annotating, and it's okay to have this iterative process, but the goal is at the end you have consistent ground truth and you don't go back and forth. Super important when you have more people annotating. So we actually saw this in action already. Lindsey, do you want to show a little bit more of that, or we
Yeah, I can
move
actually
on?
explain it a little bit more. Yeah, we can actually see a little bit more if we have just a second. So I wanted to show this
quick,
in action.
and then we move on to the.
Yeah, we'll do this really quickly. So what we have, not only do we have the annotation assistant, which is going to go find low confidence regions based on the last training region, we also have a verification tab. And I recommend that people use this mostly to find their own errors in their annotation. So it's okay, like Alex said, it's okay to be wrong, but it's not okay to keep being wrong in your annotation. So what you can do is you can have the computer sort of go to every single one of those training regions and find exactly where you made mistakes. It's going to calculate the rate of false positives and false negatives. You can also get your sensitivity, precision, and F1 scores out of this, which at this point is not as valuable as just seeing sort of where are the errors. So the computer here, you see that the sort of colorful overlay that we've seen before. And so what that's showing is after the last training round, what the computer thinks these classes are versus what I've annotated. And so it's going to tell me, and it's going to calculate in this region, for every annotation I gave, it's going to calculate the numbers of false positives and false negatives. So if I turn back on those analysis results, I can see that I definitely messed up training this region. I labeled a red blood cell here, a non-infected red blood cell as an infected one. I've labeled, so I labeled that incorrectly, and this was on purpose, of course, for this webinar. And then here, I've forgotten to give a label for this red blood cell here in green. This should have had a green label. So there's a couple of things. We can actually switch the class here quickly. Let me go to red blood cell that's unaffected. You right click. We have a lot of like hidden menus and extra tools that you can use that'll really expedite the process. So you don't have to delete it and then re-annotate it. But you would just simply, so the computer basically did all these calculations for me inside of this training region, and it specifically took me here. It's pre-sorting high to low error rate. You can also sort by your false positive, your false negative if you need to. But really the goal is that you go back, and it's completely okay to make mistakes. You just got to correct them, right? So now this is corrected, and my ground truth is going to be more consistent in the next round of training. So that was sort of the first step. So how to correct incorrect annotations. The computer is going to automate that with verification. But Alex also mentioned that visually, once you activate your results, what you're going to want to do is it's more valuable to give the computer information where it's failing. So wherever the AI model failed after the last round of training, based on those initial annotations that you gave, that's exactly where you go and you add a new annotation, for instance. So here, it's failing in the false positive direction. It thinks that this is a red blood cell. It's not. So what I want to do is actually give it the correct label. So I'm going to go ahead and label that, and then you give it a training region. Oh, sorry. So that the neural net knows where to go in the next training round. Apologize, here we go. And then we can actually take this label off here. So basically, the computer now has a new area to go to where it's actually going to get the results correct after this next round of training. And like Alex said, it's kind of a, you give the computer a little bit of information, you give the platform a little bit of information, you give it a little bit of time to learn, and then we basically let it show us not only where we made mistakes using verification, we let it show us where it's failing on the last training round, where it needs more annotations because we didn't include, maybe we didn't include that feature variability. And humans, humans don't realize a lot of times how visually biased we are towards certain features. So the computer is going to help, keep you level there. And also the annotation assistant tool, that's going to take you specifically to regions in the slide where the platform wants more information before the next training round. So all of these things are not only going to help you improve your annotations, but correct ground truth inconsistencies and show you exactly where the work is needed. So that I can actually go back here for Alex.
Yeah, this keeps us in check. It helps also, you can just start working and not worry about, oh, I know I'm not consistent enough. I annotated a little bit different in the last slide. You're going to go to this tab and it's going to show you. So last thing that we're going to talk about, and we're only going to touch on this one, and we're going to send you to a resource about this, a more detailed resource, is that no one size fits all for pathology problems. Different pathology problems require different image analysis solutions and annotation approaches. And what do I mean by different image analysis solutions? I am referring to computer vision way of solving problems. So we had this example where we wanted to localize and delineate as well as quantify the area of those tumors. And we annotate it in a certain way. And Lindsey mentioned this is semantic segmentation. Semantic segmentation is the way computer approaches this problem. But if we're doing this, we are not counting. This is just to detect the regions and to quantify the area. If we want to count, then we are using so-called object detection. And if we are doing semantic segmentation, we're not doing object detection. If we're doing object detection, we're not doing semantic segmentation. So you need to think about what your pathology problem is and then use the correct way of solving it with the correct, it ties to the setup of your project in Aiforia or whichever software you're using. But basically the concept is the important thing. But obviously there are problems in pathology where we want to have both, right? What do we do when we want to have both? When we want to have, like, let's say each red blood cells delineated or some other cell delineated, we want to count them and we want to know the area and we want to know everything. There is a solution for that as well. Like in this case, we want to count this cell. We have this object detector that we already know from the previous project, but we want to also delineate it. We want to know the shape, we want to know the area, maybe there is some hypertrophy, some other processes, shrinkage, whatever. We want to know the area and we want to have both. And this is called instance segmentation. And there is an option to do instance segmentation to have it all in Aiforia. And you would ask, why can't we like always have it all? Basically, it's easy for us to see. It's not so easy for computer to see. So each of these tasks requires some computational resources. And if you don't need to use all the resources, then just don't use them because it's going to slow down your project. It's going to make it more expensive. So basically, the previous knowledge which computer vision approach to use for your pathology problem is going to guide the setup of your project and is going to make your analysis more efficient. And if you want to learn more about that, I'm going to send you to another resource. I'm going to send you to my blog and to my YouTube channel where we have a video about what instance segmentation is in tissue image analysis, and we have all the examples of other ways of detection of semantic segmentation with a video showing this on a different project in Aiforia platform as well. So I invite you to visit Digital Pathology Place or the YouTube channel of Digital Pathology Place and see that video. Anything we want to, do we have time to see anything, this instance segmentation in action, or?
If we give 10 minutes for questions, yeah, we can take two minutes to look at instance segmentation.
Okay, let's look at instance segmentation.
Yeah. So let's actually look at that use case. So this is a pretty interesting use case. I can actually show you how it works in the platform. So we do support multi-channel images. You can actually toggle on and off the color channels individually. Here I just have the DAPI in blue active, as well as the map2 here in red. Really the goal of this entire project, yeah, and I can change the color here. The goal of this entire project, though, if I show here, was to actually get the intensity, the grayscale intensity of this green channel, which is the TDP43, to look at semantic to nuclear translocation in various conditions. So what you can do here, this is also a different type of AI model. We have multiple trained neural nets working sort of hierarchically and simultaneously. But in the first neural net, we wanted to train not only, the important thing for this investigator was to be able to detect the variable sizes and morphology of these cultured cells. So you can see the morphology of each cell is quite variable. And so instance segmentation allowed us to separate individual events and get that variability in morphology. So if it's just regular semantic segmentation, if there's two cells really close together, that annotation style will actually merge the results. And so we want to pick out individual events. If you just do object detection, you're losing information about the morphology in the surrounding cells. So here we're using instance segmentation to try and capture that. And with underneath that, we have another layer that does regular semantic segmentation. So it's actually a pretty straightforward annotation style. It's basically, it's everything that you've already seen, but it's just two of them at the same time. So here, I'm just gonna label this. You basically, it's first an object detector, you activate that object, and then you have the ability to add in your actual instance for this cell body compartment. And it's really that simple. So yeah, I think we can go into questions at this point. We can look at more examples if anyone has specific questions, but I think that's sort of it as far as this portion of the presentation.
Okay, thank you. I am going to look through some questions now. So firstly, how would you go about giving annotation trainings to other people if it's not possible for a pathologist to annotate?
So yeah, I kind of faced this problem. I talked about it a little bit. What you do, what I have done so far, you make a project team, you explain the concept. Let's say, let's take this red blood cell project as an easy example. You explain the concept, what is your class, what we're annotating, you let everyone annotate, you train the model, you go back together. The important thing is to go back together and look where the mistakes are, because the mistakes are going to be coming from different people, and they're going to see what's happening there. Good practice is to keep a ground truth document, which I know there's an option to have this in Aiforia as well in your project, like a notebook, a ground truth document where you put screenshots in, and as you evolve in the project, you put more screenshots and you address the problems that you are encountering. So there is part of this training would be basic training on understanding what structures we are annotating, and then meeting consistently to review the ground truth so that at the end we have a consistent ground truth set to train the model. I hope that answers the question.
Okay, thank you. Another question is, how do you validate the AI model?
Yeah, so that's a good question. So Alex can probably speak more to different methods of validation, but at least with the Aiforia platform, there is a validation tool. So just like you saw the verification where it's going to all those different training regions and it's comparing how the AI performs against the original input annotation. We also have a tool where we can invite external validators. They only get invited to that project. They are allowed to annotate. They provide their annotations and validation ROIs, and then you get this pixel level calculation that the computer will do for you. And you're not only comparing how does the AI perform against individual human validators and as a whole, but it's also going to show you what's the inter-observer variability. So we consider a model, an AI model validated or valid and ready to use for image analysis when it's not inferior to the inter-observer variability that we see among at least 3 to 5 individual experts. We recommend at minimum you have 3 people so that you're statistically powered enough to make that interpretation. But that's basically how you would do it in the platform. In the future, we might have another webinar and just show you how to validate with the validation tool. But for today, it was sort of out of the scope of today's talk.
And we actually, with people from Aiforia team, with Tom, and Charles River team, we published a paper on validation or on qualification of image analysis models. It's not only specific to AI and deep learning, it's for how to validate image analysis results. And it is both for AI and non-AI, and there's a process outlined. So if you embed this process in your model development, and we will maybe provide this resource, the link to it in the follow-up emails, we could do that. So there's a process, and incorporate this process into your development, and at the end you will have something that is validated or qualified or whatever you want to call it, ready to use, and you're sure that you can use it and that it's actually detecting what you wanted to detect.
All right. We have an annotation assistant question. What is the logic behind the annotation assistant? Is there a deep learning model behind this assistant? And if not, how does it propose the annotations?
That sounds like a computer science engineer question. From my understanding, it's basically finding regions of the slide where it doesn't know how to make a call. Like, is it the red blood cell class? Is it the white blood cell class? And it's going to take you and show you that region specifically. As far as the details behind how it works in the back end, I personally don't know, but Alex, do you have any suggestions?
I don't know the back end either, but basically this is the model that you're developing, and your model, what you're feeding into it, your model is showing you its performance, and it's converting its results into annotations. So like Lindsey showed that, oh, you can mark the green cells and say, oh, I want this to be my annotations. So it basically analyzes the whole slide. You don't have to annotate everything, but you can convert every region that you think is suitable into annotations. So it's like going back between results, the masks, and annotations. So between your input and output, and if you like the output, you take it as your input and use for, or you correct, right? But basically it's the same model that you're developing. So it is a deep learning model behind it because you are developing a deep learning model.
Mm-hmm.
We have a training area question now. So someone is wondering how large the training area should be.
That's a good question. And I think that there's some interplay between the training area and some of the pre-training hyperparameters, like the field of view. Basically, the field of view is how magnified, the magnification at which the neural net's learning. And so it's not a straightforward question. Actually, the answer is different for every set of images. But like we showed, so we showed kind of two very different types of use cases. We showed the lung tumor, which was a whole slide image, and then we showed the smears with the cytology example with the malaria, the red blood cells. And so we recommend, again, that you keep your training regions manageable. If you're so zoomed out that you're losing details about tissue borders and things in your annotation, it's probably too big. But if you're so zoomed in that you're losing pixel information maybe about the surrounding areas, you're probably too magnified. And when you're actually making that annotation. So what I sort of showed was kind of a reasonable magnification where you're going to want to be annotating. And that's where you can physically, as the human and as the expert, like developing this ground truth, right, that you can actually annotate. So there's no shotgun answer to that. Like, it's not one size fits all. It's each project is different and each ground truth is a little bit different. So it's keeping the goal though is to annotate in a way such that you are reducing your own errors, that you're not including background pixels in a true positive annotation, because that's very confusing for the neural net. I don't know, Alex, do you have any recommendations here?
It's usually, so from what I have seen, your training region usually does not exceed the size of, like, I don't know, for object detector, it's going to be more, like, let's say you would have a training region where you have 10 or 15 of those red blood cells. So it's not, the size of it is slightly larger than the annotations you make. Mm-hmm. So that exactly for the reason to keep everything in the training region labeled. If it's too big, there is a risk that something is not going to be labeled. Like, let's say you take 1,000 red blood cells, the risk that there is going to be 5 that you didn't see in this training region is a lot higher than when your training region only have 5. So it's better to make 5 regions of 5 or 5 regions of 10 where you really have everything annotated than a big region where you miss something.
Okay. I think we'll take one more question. So, can we use the annotations made on a slide used on other slides with similar tissues or similar stains?
Yeah, that's a good question. So we do have the ability to move annotations in between AI projects. So an individual AI project is an individual AI model where you've seen a couple of examples here. So we do have that ability. What you would need to be careful about though is, so if you're stripping a slide and then restaining it, is that the tissue coordinates have not somehow changed. And so you can move the annotations. You might need to make minor updates just due to natural variability that happens when you're stripping and restaining. But theoretically, yes, you could do that. But we also have something that we haven't even mentioned yet today. We do have a co-registration portion of our platform that we call image match. And so if you have an H&E on one side and IHC on the other slide, you can actually run basically 2 AI models in parallel and get distribution metrics between the 2 stains, for instance. So that's a really interesting part of the platform, and we could talk more about that later.
You
Alex,
have one other thing that might be helpful here. It's not going to be like for the same slide, but let's say another project, the transfer learning.
Mm-hmm.
So you can kind of take the model that you have, apply it to different slides, or in my case, that would be like different species. And then you can use an annotation assistant to generate if it performs well. If it doesn't perform, it not always performs, then you have to start from scratch. But if it performs well in at least part, you can use it as annotation assistant and generate annotations from the transferred model and then refine it.
Yeah, that's a great suggestion. So we do now have transfer learning capabilities and external nets. So what you can do is you can take an already trained AI model, you can apply it to those images, have the platform generate suggestions, basically annotations, and you can either use the annotation assistant or the converter tool to convert all of those.
Confusing.
Yeah, it's okay. Conceptually, it does similar things, but yeah, so that's actually a really great recommendation, is that you can, and then also with transfer learning, you can also kind of mix and match AI models and update them. So let's say you have a new data set that came in that was scanned and you need to do a little bit of training. You can actually retain whatever percentage of the original neural net that was trained. You can retain 50%, and then you just add a couple of new annotations to the new slides. You retrain the other 50% of that neural net on the new slides, and now you have a model that's going to generalize across all of those images. So there's a lot of advantages to being able to use transfer learning as well. And that's a pretty advanced topic. So we might have a whole webinar in the future about that as well. But yeah, I think that's kind of my only recommendations there.
Okay, I think that's all the time we have. We will be sending a recording of the webinar around via email, along with any unanswered questions. But also don't forget, you can also book a customized demo for free on our website on the link you can see, or email us at contact@aiforia.com. Thank you, Lindsey and Alex, for this webinar, and thank you for everyone who participated. Goodbye.
Thank you very much. And I invite you also to join our newsletter at Digital Pathology Place, if you haven't already come from there.
Thank you, everyone.